Hello everybody, this is chapter 15, part A. So we're going to split this up into two parts. We'll do the visual system first and then the auditory system, or vestibular cochlear apparatus too, tomorrow. Okay, so the special senses. So we're going to start with the visual system. So we've got the eye. The black part is called the pupil. The iris is around the pupil, and the sclera is the white around the iris. There's blood vessels that come in here called conjunctiva. When there's a conjunctivitis infection, it's of these blood vessels. They get a little bacteria in them and make the eyes red, and a lot of vasculature, neovasculature happens where new blood vessels grow. The palpebral fissure is the opening of the eye, a slit. Okay. So there's the lacrimal duct, or lacrimal gland, I should say, that is above each eye. And this is what's responsible for producing tears, or the wetness of the eye. And these tears then flow through excretory ducts of the lacrimal gland across the eyeball into the lacrimal canaliculi. And then it drains down this lacrimal sac into the nasolacrimal duct and into the nose. This is why when you're crying, you also have a runny nose, because that's where the drainage goes. There are a number of muscles that we didn't cover under the muscle section, but we will cover them now, that move the eye. So the first one is the superior oblique. This one has its own cranial nerve 4, if you remember, because it's got this trochlea here, this little pulley. So recognize the trochlea, and you can recognize the superior oblique muscle. It's responsible for turning the eye laterally and upward. And then inferior, there's an inferior oblique, as well as rectus muscles, and these are just very straight muscles. Rectus means straight. So you've got superior rectus, the inferior rectus, the lateral rectus, and the medial rectus, which is not seen in this picture. Okay, just another view. We've got the superior rectus, so this is from a superior view, and the medial and lateral recti there, and then below we can just pick up a little bit of the inferior rectus. And then of course we see the superior oblique going through the trochlea. Okay, so if you want to move your eye, this is the right eye, so if you want to move it out, the lateral rectus contracts and moves the eye out laterally. To move the eye medially is the medial rectus. To move it up is the superior rectus, and to move it down is the inferior rectus. Then the inferior oblique, when you pull on this will rotate the eye and point it downward, so downward right. And then the superior oblique would when it's contracted, it would also move the eye upward. This is more like rolling your eyes. And this little table shows the controlling cranial nerve with each muscle. So the oculomotor, or cranial nerve 3, innervates the medial rectus, the superior rectus, the inferior rectus, as well as the inferior oblique. Now the trochlear, or number 4, innervates the superior oblique, and that's it for that one. And then the abducens, number 6, innervates the lateral rectus. So lateral rectus moves the eye laterally. The medial rectus moves the eye medially. Superior elevates the eye. Inferior depresses the eye. The inferior oblique elevates the eye and turns it laterally, whereas the superior oblique depresses the eye and turns it laterally. Okay, let's take a look at the eyeball itself. There is a posterior segment which is filled with this vitreous humor. There's an anterior segment which has aqueous humor, a very, very similar fluid. And in between is the iris, the pupil, and the ciliary body. Now the ciliary body is what creates this humor, both the aqueous and the vitreous humor in the eye. The outer covering of the eye is called the sclera, and it goes all the way around. And then there is the choroid, another layer beneath that, and then there's the retina. Now the retina is made up of rods and cones, and we'll take a look at the retina in a little bit. Looking at the posterior of the eye, there are a couple different features. Firstly, where the optic nerve exits the eyeball is called the optic disc, and this is your blind spot, so you can't see. This is a part of the retina that is non-functional. There are no rods and cones at the optic disc, so that's your blind spot. And then there's the macula lutea, which contains the fovea centralis. And this is your point of focus where you have the sharpest vision and the most sensitive area of the retina. Okay, here's another picture showing the vitreous humor in the posterior segment, the aqueous humor in the anterior segment, and the lens. Now the lens is held by the ciliary body and ciliary processes, which will, there are muscles that will contract or relax to modify the lens shape. And you can see the different layers of the eye. Okay, so let's talk about constriction and dilation. The eye is innervated both by the parasympathetic and the sympathetic system. Now, the parasympathetic innervates these sphincter pupillae muscles that are circular, and when they contract, you get closing of your pupil, where the size decreases. And this goes along with your rest or digest. You don't need to worry about taking in a lot of light when you're resting and digesting. So for the parasympathetic stimulation activates the sphincter pupillae muscle, which is going to constrict the pupil. Now the second muscle is called the dilator pupillae, and this is activated by the sympathetic system. So regardless of the sphincter pupillae, if the dilator pupillae contracts, you're going to get dilation and increased size of your pupil. This is going to let more light in for your fight or flight, make you more aware of your surroundings, make you more sensitive in the dark. A lot of different adaptations there. So for sympathetic stimulation, you're going to contract your dilator pupillae muscle, which is going to relax the and make the iris bigger. So the path of light goes through the anterior chamber, or through the sclera, through the anterior chamber, into the lens, excuse me, and then into the vitreous humor. The light then shines on the retina in the back of the eyeball, and in the retina are a number of cells. These cells, there are actually 6 different cells here. There are rods and cones. Rods are depicted in blue here. Cones are depicted in gold. So the rods detect white light, and the cones detect colored light. Then there are bipolar cells, which are second-order neurons. And by the way, these rods and cones are embedded in the retina. So half of their body process is embedded in the retina. And you see they synapse with these bipolar cells, which are second-order neurons. Then we have an interneuron called the amacrine cell, and then ganglion cells that are inhibited or activated by the bipolar cells, which then send the signal down the optic nerve. So the pathway of signal transduction is the opposite of the pathway of light. So light comes in, bounces off this retina, is absorbed, activates these rods or cones on the very end of the retina. And then signal transduction is going to go the opposite direction towards the optic nerve. Here's another picture, this time a real picture, showing the choroid, the cones and rods, bipolar cells here, and then the axons of these ganglion cells as they emerge into the optic nerve. Okay, so this is a nice picture of the back of somebody's eye. There's a posterior wall, also called the fundus. And you can see a number of features. The optic disc, that's going to be your blind spot. And then this pigmented area is called the macula lutea. This is where the fovea centralis is located, or the portion that has the highest density of rods and cones that are sensitive to light. Okay, let's talk about the humors of the eye. So the aqueous humor in the anterior chamber forms by filtration from capillaries of the ciliary processes of the ciliary body. And the aqueous humor flows from the posterior chamber to the anterior chamber, and then is resorbed back into the venous system where it's recirculated. And so we can see it starts off at the ciliary body. It then moves from the posterior chamber into the anterior chamber and out this venous sinus. Okay, so one of the problems with vision is development of cataract. This is with usually with old age, this develops. But the cataract is a cloudiness of the lens itself. So just the lens, it develops these proteins that misfold, they aggregate, and they get in the way of the light, and the light is unable. You can see on the left eye here that you can see right through the lens. You can't see the lens because it is transparent. But on the right eye, the lens is cloudy. And so the light that comes in bounces around. It just doesn't make it to the retina, and so all you see is white light, maybe a lot of fog. So this is the lens itself that becomes cloudy with these protein aggregates. Okay, let's look at the electromagnetic spectrum. So the EM spectrum goes from radio waves to gamma waves, and in between is the visible light spectrum from about 380 to 750 nanometers. This is your visual spectrum. We cannot see infrared or ultraviolet, but we see everything in between. Now, if you go towards your blues, there's going to be increasing wavelength. Go towards your reds, there'll be decreasing wavelength. Okay, so the rods and cones absorb light at different wavelengths. So the rods absorb light around 500 nanometers, a pretty broad spectrum. And then you have three cones. You have blue cone, green cone, and red cone. So those are your primary colors of light. And so each cone would detect a different wavelength. Okay, we also have to talk about refraction, since we do have this humor in our eye. It's a different medium than the air, and thus when light comes into the humor, it is bent a little bit. And this is called refraction. And so this is showing a laser light coming in to this medium. This is whatever we're looking at. It is more dense than the air, and so the light is bent temporarily until it comes out of the media, and then it resumes its regular course. This is how prisms work and how rainbows work. Okay, so the lens we have is a convex lens, and you're just going to have to learn the shape of the convex lens. Okay, and when the light comes in, there is a focal point, and then the image flips, and the retina would be right here. So the retina would pick up this flipped image after the focal point. If you have 2 points of light, you can see that how the image flips, and so it'll start at the top here. The light goes straight through the lens, but it will bend in other places. And so the end result is the image on the retina is inverted. It's upside down and reversed. So focusing depends on the ciliary muscles that are attached to the lens. And these muscles are arranged like a sphincter, and when they contract, instead of pull on the lens, it loosens the lens. And then when they relax, the lens tightens up. So it's a little bit opposite of what you might think. So here we show focusing on distant and close vision. So let's start with distant vision. So distant vision, the lens would flatten, and you would, when you adjust your vision to the distant, you can feel your eyes strain just for a second while these muscles reaccommodate the distance. So the ciliary muscle is relaxed, and this flattens the lens because of this tightened ciliary zonule. For close vision, the ciliary muscle contracts, which loosens the ciliary zonule, letting the lens naturally do its bulging. This would be like the natural lens shape would be bulged. So with the relaxed eye, you can read up close. When you look far distance, you need to contract your, you need to relax your ciliary muscle to pull on the lens to change the shape. So once again, for distant vision, it's relaxed ciliary muscle, but this pulls on the lens to flatten it. For close vision, the ciliary muscle contracts, which loosens the lens, allowing it to go back to its bulging state. So there are different types of sighting. Emmetropic is the normal eye, which can see nearsighted and farsighted without accommodation or correction. And you can see the light comes into the lens. It then is bent into the focal plane, which is going to pick up in the retina. The myopic eye is nearsighted. Okay, so the eye is too long, so you can see nearsighted, but you cannot see farsighted. And since the eye is too long, the retina is behind this focal plane. And with the corrective lens, actually a concave lens, to correct for this over-convexness, the focal plane is then appropriate. Hyperopia, or farsightedness, is when the eyeball is too short. And you can see far, but you cannot see near without accommodation. And this involves a convex lens, which will then move the focal point forward So emmetropic would be the normal eye. Myopic would be nearsighted, and hyperopic would be farsighted. Okay, so the photoreceptors of the retina come in two flavors. They're rods or cones, and this outer segment is embedded in the retina, and the rest of the rods and cones are exposed to the humor. And we've got our synaptic endings, where we would then synapse with an interneuron. So if we compare the rods and the cones are non-color vision. They're highly sensitive, though, and they function in dim light. But there's low acuity, which means that there are many rods that converge onto one ganglion cell. There are more rods than cones, and these are mostly in the peripheral retina, so not in the fovea. The cones have low sensitivity, but they function in bright light, but they have high acuity, which means that they're each cone is going to have its own ganglion cell. But they're less numerous than the rods, and you would find the cones in high density in the fovea or the central region of the retina. So the photoreceptor is rhodopsin. This is the visual pigment in rods. As well as retinol and opsin. So these are molecules that make up the photoreceptor. So the formation of rhodopsin starts with 11-cis retinal, which is derived from vitamin A. Vitamin A helps you see at night, helps your vision. So when 11-cis retinol is combined with opsin, it forms rhodopsin. And a light absorbed by rhodopsin triggers a series of steps in which the retina changes shape and eventually releases opsin, and then enzymes will convert the retinol to its 11-cis form. This requires ATP, and this leads to a signal transduction pathway. So if we look at the events of phototransduction, the first event is retinal absorbs the light and changes shape. This activates a G protein. It's a GTP binding protein called transducin. And this whole pathway allows for amplification, so you can have activation and then amplification of your response. So once transducin is activated, it then activates what's called a phosphodiesterase, which then converts cyclic GMP into GMP. cGMP levels fall. There are ion channels that are sensitive to this process that then close, and the result is hyperpolarization because you're closing your ion channels and the cell hyperpolarizes. So once again, the retinol absorbs the light, it changes shape, activates this phototransduction where 11-cis retinol moves the activation of this GTP binding protein called transducin. Once the GTP binding protein is activated, transducin then activates phosphodiesterase, which converts cGMP to GMP. Now cGMP activates a number of ion channels, so when you convert it to GMP, you inhibit these ion channels, so you're not going to get sodium and calcium to move in. This is going to then hyperpolarize the cell or inactivate it. So if we take another look, so we start at this cell body, which is embedded in the retina. And this is going to be in near the fovea. So its cGMP gated channels are open at rest. This allows influx of ions, and the photoreceptor depolarizes. Then voltage-gated calcium channels open in the synaptic terminal, leading to neurotransmitter release. This then leads to inhibitory postsynaptic potentials in the bipolar cell, and hyperpolarization results in this bipolar cell. So you've got activation of the photoreceptor, the rod, that then sends an inhibitory neurotransmitter to the bipolar cell. It hyperpolarizes the bipolar cell, leading to decreased excitatory postsynaptic potentials in the ganglion cell. And so this leads to no action potentials along the optic nerve. Now in the light, the CGMP gated channels close because we've undergone our signal transduction. So the photoreceptor here hyperpolarizes. Now this lack of inhibitory neurotransmitter released results in depolarization of your bipolar cell and activation of your ganglion cell through excitatory postsynaptic potentials that then lead to optic nerve action potentials. Now I do want to say that the neurotransmitter that works in the dark is called glutamate, and so glutamate is shut off in the light. So in the dark, photoreceptors are depolarized, leading to glutamate release. In the light, glutamate release is inhibited, and this inhibition is of an inhibitory current. So therefore, your cells, which are normally active, activate and send action potentials in the light. Okay, so let's look at the visual field. So the optic nerve runs out of the posterior of the eye. It crosses at the optic chiasm, stops off at the thalamus, where it receives input from both eyes, and this builds representation of the contralateral visual hemifields, which means that the right side of the thalamus would map out the left eye, and the left side of the thalamus would map out the right eye. And then there are processes that proceed to the primary visual cortex, where it is mapped. Okay, that is the visual field in 32 minutes. All right.